Good afternoon, everyone. A very warm welcome to all of you. I am Garima. I am Assistant Professor in the Department of Biochemical Engineering and Biotechnology, and it gives me immense pleasure to welcome you all here for the Professor T.K. Ghose Memorial Award and Lecture 2025, organized by Department of Biochemical Engineering and Biotechnology, IIT Delhi, and endowed by Professor T.K. Ghose, a pioneering researcher who laid the foundation of biochemical engineering research in India and chaired our department till 1985. Today, we will honor the outstanding PhD research contributions of the young scholars of IIT Delhi in the area of biochemical engineering and biotechnology. As part of the ceremony, we also have Professor T.K. Ghose Memorial Lecture, which brings to us an eminent speaker who will share insights from the frontier areas of biochemical engineering and biotechnology. I now invite Professor T.R. Srikrishnan, Senior Professor of Department of Biochemical Engineering and Biotechnology, IIT Delhi, to introduce the department and the Professor T.K. Ghose Memorial Award to us.
Good afternoon, everyone, and welcome to the 5th edition of T.K. Ghose Memorial Award Ceremony and Lecture. Professor T.K. Ghose, Tarun Kumar Ghose, fondly called by his colleagues and students as TKG, born on September 1, 1924. So if he was alive today, he would have been 101 years old. So he was born in Bengal. He was a visionary leader, visionary because he was the person who initiated the academic program called Biochemical Engineering in our country. First at Jadavpur University, followed by HBTA Kanpur, then he moved on to IIT Delhi. He joined the Department of Chemical Engineering at IIT Delhi in 1969. And that time they had a division of biochemical engineering within the chemical engineering department, which separated out and became a separate center. We call it a Biochemical Engineering Research Center. It used to be called BERC. This was started in 1974. And later on, this department, this center became the Department of Biochemical Engineering that we have today. Professor Ghose played a key role was the editor for the journal, very well-known journal, Advances in Biochemical Engineering and Biotechnology. And he is a highly decorated scientist, so many things. I am not going to read through all of this. He is a very well-known person in the area of biochemical engineering in our country. And he started the department. Of course, when he was there, it was actually a center, BERC, which became a department slightly later after that. The chronological order in which the department evolved, initiated by Professor Ghose. 74, we had the BERC, Biochemical Engineering Research Center, which started with an MTech. There's a 2-year MTech and a PhD program. Later on, in 1989, we started the 5-year integrated MTech program, like in other few disciplines, electrical, computer science, and chemical. Along with them, we also had started this program, 89. And as part of this, the center also got upgraded to a full-fledged department. In 1993, it was approved, it was given the status of a Department of Biochemical Engineering. In 1998, the integrated MTech program was slightly modified and it became a dual degree program. Earlier, we were awarding an MTech degree after 5 years. As part of the dual degree, the structure is that the student gets a BTech and an MTech degree at the end of 5 years. In 2014, we converted this dual degree program into a 4-year BTech program, which is what we have right now, the undergraduate program. We had only about 30 students admitted as dual degree, but when we started the BTech, it became 60 to 65, and now we have approved strength of 70. In 2022, we also started a 2-year MTech program, which is most recent program of the department. So currently, we have a 4-year BTech program, 2-year MTech program, the MS by Research program, as well as the PhD program. And we have a faculty strength of 20 now, one of the highest number if you look at the last few years. There was a time when we were only 15, 16 people, even though we had a BTech, MTech, MS, as well as PhD program, total faculty was only 15, 16. Now we have 20 faculty members, and of course the number is already given, assistant professor, associate professor level. We have a large number of postdoctoral fellows joining us, as well as project staff who work in various sponsored research projects which are under operation, and some technical staff. Coming to the research areas that our colleagues, me as well as our colleagues in the department are working on, broadly we can put them into 4 baskets. One is on bioprocess and metabolic engineering. Second one on systems computational biology as well as theoretical biophysics. A third one focusing on environmental biotechnology. And the fourth one on molecular biotechnology of diseases, molecular biology of diseases, diagnostics as well as bio-nanotechnology. We have extremely prolific publication list. We had been doing extremely well looking at the overall publication of IIT Delhi, because you know that the ranking of IIT Delhi, whenever it has been high, one of the biggest contributors has been our publications, and our department has always been in the forefront of the publication history of the institute. Also a lot of patents also. Now, as part of this award ceremony, the memorial lecture as well as award ceremony, instituted by some of our alumni, majority of which who are also directly students of Professor Ghose. This is the 5th edition of that. There are awards given to best PhD thesis in this area. It need not be from our department, it is from other departments, those who are working in this area of biochemical engineering as well as biotechnology, as well as a lecture by a well-known scientist from invited lecture. So today, this year, we have 2 students who are getting this award. One is Dr. Shantanu Banerjee, who did his PhD in chemical engineering, our chemical engineering department. And second is Nitika. She also did the PhD from chemical engineering department. And today, we have an eminent scientist among us to talk to us on this occasion, Dr. Taslim Arif Said. He is currently the CEO as well as director of CCAMP, the Center for Cellular and Molecular Platforms, Bangalore-based institution under the Department of Biotechnology. So he will be talking to us. So this is the overall introduction of this program, and I would like to hand over to Garima to continue with the proceedings.
Thank you, Professor Sri Krishnan. I would like to now invite Professor Rangan Banerjee, our esteemed director, IIT Delhi, to share his opening remarks and formally inaugurate the ceremony.
So good afternoon, and it's indeed this is an important occasion for us for DBEB. It's always good when we remember the original faculty members who actually helped set up the institution and the department. And Professor Ghose in 1974 laid the foundation through the BERC, and I'm so delighted that his students and his colleagues have instituted this award. So and the other interesting fact is that biochemical and biotechnology is not just restricted to DBEB, so that both the awardees happen to be outside DBEB. So that's also interesting. And so I think going forward, we have a lot of expectations from DBEB in terms of biochemical, biotechnology, integration, looking at healthcare, looking at waste management. The Professor T.K. Ghose Award is given to research students, and we congratulate the awardees. We are also delighted to have Dr. Said, who's the CEO and the director of CCAMP. Not only is he a prolific researcher, but he's also been involved in translation and in looking at innovation and looking at the innovation ecosystem. So I'd like to extend a warm welcome to Dr. Said and congratulate the awardees, and hope that DBEB can actually fulfill Professor Ghosh's vision, and we can really take this forward and enhance our impact in this domain. Thank you.
Thank you, Professor Banerjee. It is now my pleasure to introduce our distinguished speaker, Dr. Taslim Arif Sayed, CEO and Director of Center for Cellular and Molecular Platforms, CCAM Bangalore, an initiative of the Department of Biotechnology of Government of India that has been at the forefront of fostering biological innovations in India since 2009. Dr. Sayed is a neuroscientist by training with a PhD from the Max Planck Institute for Brain Research, Germany, and postdoctoral work at the University of California, San Francisco. He is also trained in biotech and innovation management at UC Berkeley, UC Santa Cruz, and the Wharton School of Management. At CCAM, Dr. Sayed has been instrumental in building India's life science and healthcare innovation ecosystem while leading national initiatives such as the India AMR Innovation Hub. CCAMP has nurtured over 600 biotech startups and fostered academic industry collaborations nationwide. Several of the startups are carrying out game changing trials focused on areas such as antibiotics, medical devices, and renewable energy. We are delighted to have Dr. Syed with us today to deliver the Professor T.K. Ghosh Memorial Lecture 2025 titled Building Ecosystems for Fostering Cutting Edge Life Science and Healthcare Innovation. Everyone, please join me in giving a warm welcome to Dr. Syed. I invite Dr. Taslim Arif Syed to please come on stage and kindly begin his lecture. Thank you. Thank you for coming. One second.
Good afternoon, everyone. It's an honor to be here for this very important day for Professor Ghosh's Memorial Award and sharing some of the work that we are doing. Great to be here among dignitaries, Director IIT Delhi, Professor Hasnain, Dr. Anurag, and many other colleagues whom I've just met. Dr. Preeti, thanks for the invite as well. What I would like to attempt to do in next 25 to 30 minutes, and hopefully if there are any questions, happy to take questions as well, in terms of what work we have been doing and get your insights on what we can do better together. I think that's the larger objective of the talk. So I'll begin with possibly just introducing the organization. Some of you may have heard of it, some of you may not have heard at all. So I'll take a point to just quickly introduce. As you heard in a very kind introduction, by the way, thank you for that. CCAMP is an initiative set up way back in 2009 by DBT. Idea was to set up an organization which would potentially play a role of a catalyst, an enabler. I don't want to, I think the department is well versed to understand the role of a catalyst across entire the scientific work they do. So largely idea was to build this catalyze capabilities across. And for me, if I build an analogy, it is like on a soccer field, it's like a midfield player, which works with both the defenders as well as strikers and helps the movement of the ball towards the final goal. So it's like an organization which attempts to work with academic researchers as well as product developers and see how the scientific movement can happen from one side of the field to the other side of the field by understanding the language at both sides and translating that science, which can be taken up by the strikers. And that's a very important thing because eventual gratification is when the scientific outcomes actually impact the society and scores the goal. And hence, it was clearly possibly felt that there was a need for an organization which could play this catalyzing role to enable things. And what way? So, how do we attempt to do that? That's a larger mission. What do we enable? Idea is to enable high-end science on research, high enable scientific science-based translation of research, so enabling research, enabling translation of research, and enabling research-based innovations. So it's a three different steps of scientific movement: enabling scientific research, enabling translation, enabling innovation. These are the three major components that we have worked over time here. And idea is that how is that coming out? What has happened through that is something that I wanted to share very quickly with you because each step is an important component for it. Let's start with the simplest one possibly, which was beginning one, which happened. But this was the genesis that happened around 2009. It started the high-end technology platforms were set up in 2010. 12, we started innovation entrepreneurship capabilities. 13, we started the translation accelerator, which was key on how the science can go forward. And then we have, over the last few years, we have built significant expansion at CCAM by the work that we are helping. And in a way, we are now in the public health programs in terms of taking science and innovations which are ready. How are we taking to state government health departments and deploying them and validating them, which is a very important ask. And of course, in 2026, we are starting CCAMP Innovation Park, which would actually take some of the aspects to the next level overall. In 2023, CCAM became the first life science innovation ecosystem which crossed the 1 billion mark in terms of the valuation, and now we are at 1.6 billion. We are hoping that some of the inflection points that you heard, where we are in the clinical phase trials and many others, this will reach 3 to 5 billion, hopefully in next 6 to 7 years. This will be very, very good for overall for solutions perspective and of course the economic value. Who we are part of the Bangalore Life Science Cluster, which possibly many of you may know about it. Along with us, there is NCBS, which is a fantastic fundamental biological science research organization, a center of TFR, InSTEM, the only federally funded stem cell research organization, and Tata Trust has funded a $70 million effort to set up Tata Institute for Genetics and Society. And that is something is there. So all four organizations together form what you call as the Bangalore Life Science Cluster, which is apparently India's largest life science research campus and something that we want to scale it up and make it double in next 5 years overall. This gives also a very good, kind of seamless coexistence of fundamental research, translation, innovation, and so on. So it's a coexistence, but it's not under one umbrella. We work together, but it's a very amazing, I think, leveraging each other's strengths, but not necessarily driven by reporting structure, which allows national mandates to each other organization and allowing us to bring talent in different roles altogether. So as I mentioned, the role has been catalyzing research, catalyzing translation towards industry technology, and catalyzing innovations at large. That has been the three pillars of stepping stones towards our larger role of being an enabler organization of life science research. The first component being the high-end technology platforms that we all are aware of, and something that we talk about. Many institutions like IIT Delhi, many institutions back home at our place are very highly privileged institutions. But they are possibly 5% or 10% of the research institutes that we're talking about. The rest of 90% institutes are not as privileged, but there is talent, there are professors, there is scientists who do not have a key tools and technologies to take the science forward. And that is actually impacts the progress of the science, right? And how do we actually, and that's a very vicious cycle because since they are not in a privileged institute, they do not have high-end technologies and capabilities to move fast and produce high-end results, which can get them funding and then do that. It's a very difficult cycle they are left in. One of the aspects of CCAM was to how do we unlock the high-end capabilities in Privilege Institute and then provide it to everyone outside. And it was very, very important. Can we actually convert availability into something called accessibility? It has been always available, but is it accessible? And that's a kind of a journey one has to do. What we have done is that we have opened everything that has been on campus across all four institutes for last 15 years and made available and accessible to researchers outside, along with high-end technology scientists who are PhDs, postdoc, but in a technology field, and it has been really fantastic. So all high-end tools which have been there in terms of any kind of mass spectrum you can think about, any kind of microscopy, flow cytometry, genomes, high throughput screening, um, analysis, structural determination through NMR, X-ray, even cryo-EM, they all have been now available to a state university, to a not-for-profit research institute, to an individual user, to industry, to Biocon, to Pfizer. Anybody can come in, they don't have to do it. So think about it, it actually, the available space, available capability, available people have been now opened up. And this has been also brought in as a technology training programs that I'm sure many institutions do. But that has resulted into nearly there's an older slide a bit, 6,000 projects have been done through this extra capacity which was available on those machines. More than 2,000 researchers have been hands-on trained. More than 600 organizations have been working with us. And nearly 300 publications have happened through that. So this has really made many talented scientists which were not in privileged institutes and well-funded institutes to actually fast forward their science and not stagnate because they were not there. And that is, in a way, kind of trying to get parity. You can never completely address it, but trying to get parity within the research ecosystem and see how one can actually do it. So this has been very good example. Now we have on this thing, we have now loaded many other institutes. So initially we started with 4 of us, the cluster organization. Now many institutes actually have come on it. The front remains CCamp, and whatever we do not have or we do not have today, we actually are leveraging other institutes on it. So many institutes are actually onboarded on this platform now. So we continue to work with 600 organizations which are top. I think most IITs have got more ISCs, ISOs, Pfizer, Biocon, Reddy's, Serum, Reliance. Everyone has been able to leverage it. And what we have been able to do is bring everyone on board and not necessarily let them struggle, because we have kind of, by walking and falling and again walking again, we have been able to bring worked on the model which is working for us, which is sustainable model, which actually is not inviting anybody to do a capital investment, but only high-end capacity which is available is being converted. So it's serving many purposes through that. So that's something we have done over time. This is first vertical, that is enabling research in across India. The second vertical is enabling research translation. And again, this was actually a response to what we saw. While we started, CCAM realized that there were really amazing science that was being done, not possibly at the scale in Western countries, and we all agree on that. There's no harm in accepting where we are. But there have been really brilliance of science in spots of brilliance of science across India, where we have publications in Nature, we have publications in Science, PNAS, we talk about JI, and many other things, JBC, and many other things which happen. But the numbers are not as many. So, but what happens to those science which actually has been done and has tremendous potential? We don't see conversion of it further. And one of the reasons that we realize that they are at a stage of the understanding of that science, that is discovery. So we took some of the science which was happening, including from here and others, and then started talking to industry and say, why this is one of the best understanding of that pathophysiology of that disease. You are the leading industry in this, sitting out of South San Francisco. Let's say I was talking about an antiviral company, right? And why would you not take it? And because they were friends, they said, this is really good science. I actually would say this is the best science in terms of understanding of this biology you've done. But unless you actually take that knowledge and build it into industry ready, that is from TRL level, that is technology readiness level 2, 3, to a validated technology, I cannot take it to my chief scientific officer. So in other words, it is like that the science is right now in the form factor, what you call it across is like this. The discovery is done, but it is like in a square. And what industry wants eventually is like a circle. And you have to kind of smoothen the edges and make into a product which is industry ready at the TRL 5-6. It's almost like you want the academia largely, not always, is standing on one side of the river after the publication. Industry is standing on the other side of the river, saying that somebody has to actually take the work from here to there. And that was a very big thing. And in a way, if we all know, right now in academia there's a significant amount of good work is happening, but it will not automatically translate because of this space. And not all academics will become entrepreneurs as well. It will be unfair to actually expect all academics to go on to do that. Industry is not able to take risks. So validation across in the cell-based assays, in toxicity work or the scale-up capabilities or other contamination issue, whatever validation you do and bring it to a TL 5-6 is something that they want for. And this is where we have proposed what we call as a translation accelerator, which actually through a national call invites academics to come and present their academic idea, which is in a discovery mode. We talk to industry. Industry says, fantastic, but can you do A, B, C, D, E? And if we consistently hear that from industry, we actually do that work and then go back to industry and saying that this is what you actually were wanted. Can we now say that this is industry technology and translate it? So over last 3-4 years, we have done it as a pilot. We have invited from all top institutes through a national call, so it's open call, anybody can actually do it. We have been able to brought out several projects which we have worked on and added that validation layer on it, the translation layer on it, industry-centric translation layer, not academic-driven translation layer, industry-centric translation layer. So we knew, or we actually had a clear to-do list from industry that this is amazing science, can we do A, B, C, D? So it becomes, for me, a de-risked, a risk-mitigated, validated technology. And we have been able to put it out to industry back. And this is something, a very small pilot we have been able to do. I don't think this scale can serve India as a whole, but this is one of the efforts to address the drop, the nosedive that was happening after amazing academic outcomes before industry can take it, because the gap was too big and it continues to be too big for it to automatically go to the next level. And we need mechanisms and vehicles which actually can plug this gap. One of the things is entrepreneurship, but how many of deep science breakthrough entrepreneurs can we actually have it today? And hence we'll need a lot of those other mechanisms to support that. So these are some of the technologies we have built over time here, which we are putting out to different things. Some of the and these are some of the academic collaborators whose original work was taken in, being translated and taken forward. Right? So this is, because this was already heavily invested with amazing scientists like here. They had already built amazing outcomes. Idea is to just talk to industry, understand the missing gap in terms of the readiness of the science, and then work on that science very categorically with a go, no-go decision making, which is absolutely brutal. And say, this is what we want to do. We will not move away from what is being asked from our translation roadmap. And work on it and then go back to them and convince them to put it out. And that is one of the effort that we have done over time. And so some of the outcomes have been, we have done some collaboration with IIT Madras, where we have given it out to NDDB as a final very big thing in animal biotechnology. In sepsis, where there is no cure, we took some of the work from ILS Bhubaneswar and put out a drug adjuvant, which has been taken by a Dutch company in Amsterdam called Survivex. And we had the opportunity to do it in the presence of the Dutch Prime Minister, who is now, of course, the NATO chief. But we also have now ability to take our science and put out internationally for space like where there is no cure like sepsis. And this is what the translation actually can do if we can constantly do it. There'll be failures, but I think all good science with potential, kind of demands and deserves translation effort. And that is something that we would like to do. Recently, this year, we have done two in the presence of course, PSA in agriculture with a very big firm called PI Industries, which is listed, very big listed company in agriculture with at least 40 to 50 countries presence for alternatives to chemical pesticides. Again, work with IBSD, Manipur, Imphal, as well as the IHBT Palampur. So CSI Institute, DBT Institute, taking their understanding first, understanding the industry, build the translation, convince them, show them across different validation trials, and then put it out. So that is a second vertical, and I'm happy to stop if there are questions. I'm happy to take as well. First vertical, enabling research. The second vertical, enabling high potential research into translation, actually, so it can go forward. And third vertical is enabling innovation entrepreneurship, which is a deep science base. So this is where we have been working across different capabilities for now 10 years, where we are able to fund them through both collaboration with national agencies. Now we are funded by international governments, international foundations to fund those amazing science-based innovation ideas. We can provide them the high-end labs, but also very importantly, scientific business, IP, and other mentorship that we can do. And of course, we have been supporting across healthcare, agriculture, and environment. Just to give you a perspective, some of the areas which are emerging significantly for us, like domains like AMR, infectious disease, maternal child health, oncology, NCDs, across within primary agriculture, there are multiple areas are there, and the environment, there are multiple areas that we are working on in this. So this is a quick snapshot of some of the innovations and the startups that we have supported over time. We have now, as you heard in the introduction, nearly 600 of those we have done, which is here. So we have supported nearly 600 startups, which we have supported. We actually funded them all through our schemes around $13 million or $14 million. As I was indicating, now they have raised $600 million against that $14 million at a valuation of $1.6 billion. And this is really amazing science story that is happening, that this is not only good science which got supported early, but now market is actually putting money in this science. We have 100 products in market, in the field, including in ICU, including 4 US FDA approved products now, including products across continents, which we have built. And this is all the journey of enabling science-based innovations that has happened. And if I may give some of the examples of those, like, so I don't know if you know, Economic Times, which is leading business daily, does something called, for last 7 years, 8 years, they do the top innovator of India across all sectors. So you would have all fintech companies, edtech companies, space tech companies, clean tech companies, all startups will be there. Out of 8 years, 7 years, it has been CCAM startups across all sectors. And that is really very good, because now there is a lot of confidence that is coming that science and science-based startups have a huge role to play going forward by people who are looking from a business perspective. And that is important, because unless there is investment in science, I don't think science-based solutions will go far in a way. And that's very, very important. So some of those are like, I'll not talk about all, but I'll give you something. Don't have a pointer, but Bugworks is the world's most advanced company right now, or effort in broad spectrum antibiotic against AMR. World's most advanced effort, right? The one that I'll not talk about, BrainSight, is working on connectomics during surgery to ensure that there are things which can be done. There are Cure.ai is working on digital pathology, and String is working on environmental work, and iStem is working on stem cell biology. I'll just give you a quick example. So this is what I started with. You know possibly about the component of AMR, and I'm sure everyone knows about it. How many of you would not know about AMR? Antimicrobial resistance. Nobody. Everyone knows about AMR. Fantastic. We all know that currently there are 5 million people dying by AMR every year, which is an understatement according to me. It is estimated that there would be more than 10 million deaths by AMR in a decade or so. And that is a huge number. It is also being seen, India is also seen as possibly the largest affected by the effect of AMR. So we have to do something about it. And the last broad spectrum antibiotic and the discovery of antibiotic, which actually had one of the biggest impact on longevity, or the last broad spectrum antibiotic was, yeah, I don't know if you, how many of you know, anybody know when was the last broad spectrum antibiotic was discovered? Starting with penicillin, but when was the last broad spectrum antibiotic was discovered? Anyone? Any guesses? Just to get you engaged, nothing else. 2012. Okay, you are only, okay, you are close by, but anybody else? Huh? Oh, early 2000. Okay, just to help you with, no, thank you for that. Just to help you with, it was early 1960s, right? That was the last broad spectrum antibiotic against gram positive, gram negative bacteria was actually the new class was discovered. It hasn't been the new class of broad spectrum, and you can imagine how much the bacteria have actually grown since then, and how in every hospital that you and I know, daily basis, people are dying of infections which are not treatable. Only thing is we're not calling them AMR. This effort started with us a few years ago, almost 8 to 9 years ago, when those scientists said that can we discover a new class of antibiotic. Fast forward a few years, and now they are actually in the phase 1 in Australia, having raised $50 million across the world with the most advanced effort, where they are working with dual approach on as an antibiotic against bacterial infections, both gram negative gram. So this will be like a magic sword that you don't want to use many times, because if you use it multiple times, the bacteria will come to know and get to adapt to it and build the resistance to that as well. But the world is actually looking at them to finish phase 1, phase 2 quick enough so they can go forward. If this comes through, without a doubt, when the world thought that we will not have new drugs from here, it will be a fantastic thing for India to develop something like this in coming years. So this is a really amazing story. They are now going immuno-oncology in terms of microenvironment of tumor as well. Second component is in Excel. This is a very important thing. I don't know if how many of you are aware of something called VAP, ventilator associated pneumonia. Good. So thank you. So VAP is, if I may say so, all of us are right now, everyday basis, and possibly I'm doing more right now. We do 1.5 to 2 liters of oral secretion, right? What do? You don't even realize it. We all are actually gulping it down, right, and putting it into our stomach and so on. What happens to ventilated patients who are also having oral secretions, but they don't have controls, and even elderly, even although not ventilated, they don't have good muscle control. So in ventilated patients, 1 out of 3 ventilated patients actually cannot control eventually the oral secretions, and it trickles down slowly to their lungs, causing pneumonia, which is called as a ventilator associated pneumonia. 1 out of 3 patients. This is global. There are globally, then what happens, nurses come, they actually aspirate it out, just like you see it at dentist's place, and then prevent occurrence of it and so on. The numbers also suggest that if the patient is elderly, 1 out of 2 patients we cannot save them, which is a major number. What they have built is very interesting work over time, is ability to build a simple device which will have a catheter and a sensor which will be near 3 sensors near trachea for humidity and moisture, which will sense oral secretion and aspirate it out and bleach it right there with a mouthwash. This has now been validated in JIPMER, we've validated in Narayana, where, let's say in one of the studies they did 30 patients, they were expecting normal circumstances 10 to have VAP or pneumonia. They only got 1 VAP in that. So that is more or less 90% reduction. This is the world's only US FDA approved device right now for prevention of VAP. There is no other device which has been approved for VAP prevention. And it is now, it was got Best Innovation Award across all Commonwealth countries, across all sectors. And now it is in multiple countries, including India. This is really phenomenal work. If I give you what was the component, who are the drivers? They were young, I would say BTech students who came out from mechanical engineering and design, and spent time in ICU shadowing intensivists to understand how their engineering and design skills could be used. Landed up through their observation capabilities on this problem, tried, tested over years, and landed up on this. This is really remarkable because without a doubt they are saving lives on daily basis by doing it. So, and they have built many other things for neonatal CPAP device and so on. If I give you a story of this, and I'll try to wrap up after 5 minutes after that, this is a story now for impact at a different level. So you know that when there is a premature birth that happens, one of the things that gets most affected neonatal mortality happens because of the respiratory issue with them. Their lungs are still not mature enough. So they have to be given a external support through called CPAP that we all heard. There's a neonatal CPAP as well. But most places in India are not conducive enough for CPAP to work. Right? There are no good neonatologists. There are no electrical capabilities. The oxygen lines are not there in many hospitals. So how do you work? So these guys have built something which actually works without a neonatologist on oxygen concentrator, on wheels, so it can actually done in ambulatory. And we deployed this machine. This is regulatory approved, by the way. We deployed these machines in all hospitals of Assam in medical colleges. I'll give you a quick data from the National Health Mission website. No, it's not our word. National Health Mission website. There's a drop of 20% reduction in neonatal mortality by a good device that understands our own requirements and solves it. So local problem, local solution, and how actually you can align that. This is really amazing data. And we were, last week I was with Guwahati with the Secretary Health there and Medical Education. We really hope to go to other states of Northeast and ensure that this actually can go further. This is really remarkable. And the story is very clear for us after 10 years of this. Local problems demand local solutions because of the direct connect of the requirements and the addressing design of the solution that is there. And that is really, really amazing. I'll skip a few more. This company is iStem on a stem cell based work, and so vision disorders is one of the larger thing, and there is something called age related macular degeneration, which happens to 200 million people, by the way. Not 1 million to 200 million people are affected by vision disorder, which is age related, which eventually they lose sight on it. The only solution that is there in the US right now is costing $600,000, one eye. If you add to, might be $1 million or whatever. What they have built is now capability to use allogenic, that is blood-based, normal blood-based conversion of cells into RPE cells, which is a degenerative component that happens here, and implant it back. And they have shown it in the cell-based data, animal data, one of the best results, and now they have finished phase 1 trial. And their data, which is giving around 15 letter recovery for a legally blind people now in phase 1, this safety data actually is best in the world, even better than Roche. Right? And they are hoping to go in phase 2 in a month or so and hopefully go further. The next phase they want to do, of course, is for lung disorders, for degenerative, and as well as neurological disorders going forward. But their last round of 10 million raise was oversubscribed, and they are hoping to do more on that. Sorry, I went back. Just to give you a perspective. Of course, Achira is a company which is doing phenomenal work in lab-on-chip platform for diagnostics, funded by now CIPLA, really do good work in immunoassays. Pandaram is a company which is only one of the 2 groups in the world which has been able to put suturable cornea, and hoping to get a larger deployment in the US and India soon. Zoomotor is a company which is using fucosylation as a base for immuno-oncology, for to be able to use maps for that, and really they are in the phase 1 trial in US FDA as we speak against cancer. In agriculture, just to give an example, they've raised more than 40 million now. A company which is doing India's world's biggest company doing ocean agriculture, both in Indonesia and Tuticorin in India, where they are growing seaweed on the surface of the ocean and using the hydrocarbon capabilities for downstream agri inputs, aqua inputs, and bio plastics and so on. Of course, they are already working with the central government and the Ministry of Petroleum for towards biofuel already, and there has been very good growth on that. Fantastic, and they are actually selling those solutions now in 25 countries. Really good work they have done. They have been funded by Tata Capital, which went through a IPO this 2 weeks back. String Bio is trying to address one of the largest issue of carbon, you know, load and the global warming that we all know about. And we all know the global warming is happening because higher concentrations of three greenhouse gases, largely methane, carbon dioxide, and nitrous oxide. And what String Bio is doing is using synthetic biology and having microorganisms who can capture methane from the source, like refineries and dairy and so on, and use it downstream. Again, one of the most decorated synthetic biology company for carbon capture in the world. And they are now using those capabilities in methanotrophic bacteria or methanotrophic organisms rather in agriculture to be able to do it in the next level of work. Really fascinating work funded by European investors, funded by Australian investors. Really good science. Something we are very, very. So these are. I just want to give you a perspective of, work that is happening in the drug areas, the medical devices, diagnostics, agriculture, and we have loads of those. But of course, you also want to hear how amazing science is happening in India. That we are all, and there is definitely a lot of connection. IIT Delhi is also in some of the stories, which I'm sure we acknowledge big time. What has happened is that over time, rather than agnostically, we have now have built center of excellences where thematic areas we are being built. So agriculture, AMR, digital health, diagnostic. We are soon to launch something called Bio for Climate. So using biological concepts for climate now, something we are going big, and of course biologics. So these are some of the companies in each of the thematic areas that we are building. Agriculture, what you see now, 70 to 80 on an average companies in each one of them, which is very important critical mass. In infectious diseases, digital health, we have nearly 100 companies. Bio for climate. Oncology, we have across in screening, diagnostics, and therapeutics. We have multiple companies that we are hoping to build on, including efforts in natural killer cell-based effort and so on. Something we are hoping to build big is on neurobiology and mental health. Something we are hoping to expand much needed space in India. Very much important space is maternal neonatal child health, something we are hoping to build big. Rare genetic diseases are very high numbers in India because of our obvious population. What we can do with early diagnostic, prenatal screening, and even going forward in vivo gene therapy that we all talk about, how it's going to work out, not only in terms of fetal therapy. Without a doubt, in 15 years, in vivo gene therapy will be, absolutely on as we go forward. Geriatrics is something that we're hoping to build on, NCDs. And doing this, now we have built global networks of best innovation hubs, like UCSF, be it now in Boston, we have collaboration, in UK with Roslin, with France, in Paris Sunday campus, Netherlands and all. We attempted to build bridges and collaboration so that we can take some of these innovations there and their innovations here, so that there is a global view of innovations, not only internal view of innovations can be done. And many of them have been possible in the presence of course, Honorable Prime Minister, but also other dignitaries. So it's a national effort that we are hoping to build on. And this all has been possible because of course, the absolutely conceptual support and the fundamental support from DBT, but many other organizations over years across national funding, national state level organizations, but also national foundations, global foundations who believed in the same thought. What we hope to do, of course, is going forward is that these are really exciting times for us to do it. The scale is nowhere closer to what we should be doing. These are just examples, to be honest. If you ask for each one of them, we should have 20 or 31 of them. For each one I spoke about, we need to have 20, 30 for us to be successful. So we need to actually work on it. The times are conducive. We are hearing about ANRF, we are hearing about RDI fund, we are hearing about many other things which is happening. The places like this are the pot where the innovations would happen, and a lot of things have happened. A lot best of the unicorns have come out of here. So I think the science and technology based innovation is great times. much can be done together. Ecosystems like CCAM are around now. So I think we can all, attempt to see where we can join hands and build solutions which can be, of course, phenomenal economic returns, but also societal solutions which you all can be proud of going forward. Thank you so much for patient listening. I'm happy to take questions. Thank you.
Thank you, Dr. Sayed, for the insightful talk. Uh, Thank you for discussing with us the journey of CCAMP and various innovations done and various opportunities we as a faculty and students have at the academic industry interface. So thanks a lot. A big round of applause for the talk.
Thank you. Thank you.
Uh, we will now take few questions from the audience. Any questions?
Hello, sir.
Hi.
Sir, I wanted to ask that as we are academicians and PhD scholars, so what do you think will be the roadblocks or difficulties if we want to translate our work into industry or commercialization, like as your experience from C Camp?
No, I think I see those as opportunities, to be honest. Uh, Once you realize that science that you are doing has some potential, it's for you to now make sure that you can take it forward. And whatever hindrances come is just a kind of a small validation that you need to ensure that if it comes from industry, is it an investment needed? There are validation infrastructure that you need. Many other things are something that you can add on, but your larger long-term goal is to ensure that your science, which is good enough to be taken as a solution in across these areas that we talk about. According to me, in last 15 years, the landscape has changed big time, and there are so many opportunities which are there. I'm not saying this is perfect still, but still the opportunities are plenty. There's a lot of good systems like us. When you visit our campus, you'll get to realize there is a buzz, which is, I'm sure, like here, which people constantly talk about what we can do more, right? What we can get together and solve some problems which are around us. So I think I won't say that there will be challenges, but there will be requirements for you to fulfill to take your journey forward. Let's put it that way. And anywhere you need support, ecosystem like us are available here. I'm sure there are ecosystems who can help you and handhold you. So the talent like you and your idea actually fulfill the potential in a way.
Thank you, sir.
Yeah, thank you.
Grafin sir. The commercialization, of course, the benefit will be ours, but there are negative impacts of that because sometimes the immediate solutions do not fill the void that as is going to be created in long term, and there are so many examples before us like plastic, like COVID medications that were given, and so many examples that acceleration has resulted in some negative impacts. So is there a corner for that also in your organization that thinks about this side of science
No,
in
very
integrated
much,
with it?
very, very much. I would say so. We have now, over time, we have built engagements with many stakeholders, including, top clinicians across different components. We have now engagements with top companies, where the innovators like you and even academics get an insights on how do we want to take it forward rather than just shortcutting it just because of the pressure that comes in, because long-term view is very important. And I think experience provides that long-term view, right? And so having people around you who are experienced in some capacity helps a lot, who have that understanding of it. And so we actually, what we do, we create environment where young innovators or even some other innovators constantly get a very structured hand-holding by this hand with very experienced, dedicated experts who come from different domains. And that actually addresses many of the shortcomings, because sometimes by listening to one person, one gets possibly affected to go towards that. But one one can actually solve those components by having a very detailed discussion on it, and that has helped a lot. So I would say that you're spot on. One shouldn't do it because once the example of the VAP or the example of neonatal CPAP clearly demonstrates that if you work hard on a problem and if you solve it, then the market will actually come and tell you that please come and how do I do it? Example that so we have deployed in multiple states now, not only Northeast, we deployed in multiple states and health minister and the health secretaries who are actually coming and saying that we need it. It actually clearly is a testament to say you have been able to build a solution which was need of the hour for the healthcare system. And that's what you need to do and have that vision and view of it, I would say.
Yeah, thank you.
Sure.
Any other questions? Okay. Let's thank the speaker again.
Thank you.
Now I would like to request our director, Professor Rangan Banerjee, to present memento to Dr. Syed as a token of appreciation from us. Thank you, Dr. Syed and Professor Banerjee. We now move on to the award ceremony for the Professor T.K. Ghose Memorial Award for Best Thesis 2025. Our first awardee is Dr. Nitika. She is currently working as Research Associate at IIT Delhi. She is awarded for her work, development and implementation of control strategies for production of biotherapeutic products. I request Professor Anurag Rathod, PhD Supervisor of Dr. Nitika, to introduce her work to us.
So Nitika is actually alumni of your, DBEB department. That's where she did her MTech. But I had a pleasure of guiding her through her PhD. Her work has been primarily on biomanufacturing, obviously, but more on continuous biomanufacturing. And the topic is that, as you know, that therapeutic products, especially biological products, MABs in particular, are very expensive. So they're not affordable even for most of Indians, right? It's really expensive stuff if you have to purchase them. And so Nitika's work has been an important component, in making these drugs more affordable. I'm really happy to say that actually partly from Nitika's work, we just got awarded by the Gates Foundation. So Gates Foundation does these grand challenges across the world. And there was a grand challenge on, ways to make MABs more affordable. So we are the only team from all of Asia to be selected in that. So we were one of the 7 finalists. So exceptional work. I'll not take long. I'll let Nitika take the podium. But I think it has been a journey. And thanks to DBEB for acknowledging it. Thank you.
Thank you, Professor Rathod. I now request Dr. Nitika to deliver a short presentation of her PhD work done at IIT Delhi.
Hi everybody. So as sir and ma'am has told you that my topic of PhD was development and implementation of control strategies for production of biotherapeutic products. As Professor Rathod has said that these biotherapeutics, they are very expensive. We have various biotherapeutics which are there in the market. They are rapidly growing, but even after that, the cost of them, it's huge. It is so huge that most of the people, especially in the developing countries, are not able to afford it. So one way of making these biotherapeutics affordable to everybody is to have some manufacturing advances. Currently, most of the industries, they are using batch mode of manufacturing, where you do one unit operation at a time, then you analyze the product, and then only after confirming the quality of the product, then you move on to the next unit operation. However, with this, there are a few key limitations. The quality of the product is not usually consistent. It has low productivity, and also it has large footprints. Thus, there are manufacturing advances that are required. One of these manufacturing advances is continuous processing. In continuous processing, you perform multiple unit operations parallelly. But since you are operating it parallelly, there is no time for you to analyze the product, because you'll need only a few milliseconds to a few seconds to analyze your product and then to make a decision if there is some error in the product and do you want to change the next unit operation or do you want to discard that product. So with continuous processing, we have a consistent product quality, and it reduces the cost of these biomanufacturing by reducing the facility sizes and by improving the quality of the product. However, since it's very fast, we need to have some control strategies that need to be in place so that we know that the product that we are making, it is of good quality. So my whole PhD thesis was revolving around developing process control strategies for purification of these molecules. So the first work that I did was to manage in-process stability of L-asparginase. L-asparginase, it can be used as a chemotherapeutic agent. However, it's majorly made in E. coli. And in E. coli, due to its reducing environment, its refolding becomes challenging. Because even after refolding, its in-process stability is a major issue. So we targeted to improve its refolding percentages as well as to improve its stability after refolding. So for that, we developed a few design of experiment-based experiments. So the first step in refolding is solubilization of these inclusion antibodies. For that, we did some one variable at a time factor optimization for its solubilization and refolding. Once we have the factors that are important for these refoldings, we performed various experiments based on Plackett-Berman-based DOE and response surface method DOE. And then we found out that there are a lot of additives that are important for refolding of this L-asparginase. Once we have identified those additives, we then found out what are the levels of those additives that are required. Because not just identifying the type of additives that are required, the amount of those additives is also important for the reaction. After this DOE, we found out that dilution factor, pH, glycine, and sorbitol percentages are important for this reaction. And then we confirmed that from the unoptimized conditions, these optimized conditions, they gave a better performance in terms of enzyme activity, as well as its stability was also improved after 1 week of storage at 228 degrees C and as well as room temperature. The next case study is to establish continuous mixing reactions. So since it's very easy to do some mixing reactions in a batch mode where you can just have it on a beaker and then you can just stir it. But it's difficult to convert these batch mixing reactions into continuous mixing reactions. So for these, we have developed an in-house coiled flow inversion reactor. And this inversion reactor is such that the D number should always be greater than 3. Modified torsion parameter should always be greater than 500. And it has to have a particular residence time, depending on what residence time do you want to give for your reaction. So with the use of inline mixture, we found that this CFIR, it gives you a better mixing as compared to it without an inline mixer or with your normal batch mixing. Because this gives you a sharper RTD curve, it also gives you a benefit that you can have sampling in between this CFIR, and you can monitor the reaction as it is proceeding. The usual setup is that you have reactant coming from one end, another reactant coming from another end, then there's an inline mixer, there's a CFIR, and then you neutralize the reaction. The first case study that we did with the CFIR is the pegylation of GCSF molecule, which is granulocyte colony stimulating factor. Again, we performed some DOE-based experiments to optimize this pegylation reaction. For this, the input parameters were pH, buffer, protein concentration, and PEG is to protein ratio. And the output parameter were reaction completion time, higher number of percentage monopeg conversion, higher productivity, and higher selectivity. Once we have this, we designed a space. There's a design space which came from these DOE experiments, which gave us that the protein concentration and PEG is to protein ratio is of utmost importance. And rest of these factors, they don't play a major role. So for a reaction that used to take roughly 12 to 13 hours, we reduced its time to 60 minutes. And we found that using our optimized condition, in 60 minutes, the protein concentration, and with this protein concentration and PEG ratio, plus if you convert it from a batch to continuous manufacturing, your reactor-specific productivity increases 7 times higher because CFIR gives you a better mixing as compared to your normal batch mixing. Another case study that we did with the CFIR is continuous virus inactivation. Since for monoclonal antibody production, we usually use mammalian cell cultures, and they are very prone for virus infections. We need to remove these viruses before we proceed forward and make a drug product. So virus inactivation is kind of crucial in those stages. So we perform pH-based virus neutralization. We reduce its pH to 3 and we keep it at a residence time of 55 minutes. And we showed that the logarithmic reduction value for CFIR as well as batch are comparable. Thus, the CFIR, it can provide you with better mixing, and also it provides you with an enabling technology that can help in providing residence time for converting a batch mixing process to a continuous mixing process. Another case study is for chromatography control. So now we have a product, but we want to purify it. So for purification, in continuous purification, usually people use multiple equipments. We have BioSMB and we want to perform all the chromatographic steps in one BioSMB so that its control becomes important and its control becomes easy because you don't have to control different equipments and integrate them. So the chromatography that we targeted was cation exchange chromatography followed by multimodal chromatography. In case of monoclonal antibodies, there are sometimes a high number of aggregates which have immunological repercussions, but they need to be removed during its purification. So usually the aggregates are removed in CEX, but there are some type of monoclonal antibodies which have higher number of aggregates, and thus an additional polishing step is needed sometimes, which is MMC. But the conditioning of these cation exchange chromatography and multimodal chromatography, they are very different. So here CEX is being performed in bind and elute mode, however MMC is being done in flow-through mode. So your monomer, which is your product of interest, will be in flow-through mode, and the aggregates that you want to remove, it will bind to the MMC. But since the pH and conductivity of both of these has to be very different, and inline conditioning is needed. So if you are running it in a batch mode, it's very easy. You just collect your product from CEX, you condition it, and then you do MMC. But how will you do it if you want to perform it in continuous manufacturing? So we found that we need to control pH as well as conductivity. And this MMC is it's very sensitive to pH and conductivity. So if pH and conductivity are variable just by slide, either your monomer can bind to MMC, thus impacting your yield, or your aggregate may not bind, thus impacting your purity. So to convert that, we did some buffer calibrations. We needed to reduce its pH from 6 to 9, so we needed to add HCl, And also we needed to reduce the conductivity. So we had to add water to control conductivity as well as NaCl. So we did some dummy runs with only buffer to find out these equations. And these equations, if you input it in Python, it will give you some calculations based on that how much flow rate of these bio-SMP pumps should be there so that your HCl, water as well as NaCl addition can be controlled, so that whatever that is coming out from your CEX is already conditioned before loading onto MMC. Once we have this model in place, we verified it using different elutions. So if we develop it only on one type of elution, it will work only on our system, but we wanted this model to be robust. So we tried it on multiple type of elutions. We tried it on linear, multi-step gradient as well as sigmoidal gradient, and we found that pH, conductivity, as well as the flow rates, they were all controlled with the Python-based software that we developed. Similarly, we wanted to see what if there is some error that is being happened in making these buffers. So for example, pH of these buffers can go wrong, conductivity of these buffers can go wrong, or there can be some error while you are running these chromatographies. There can be some leaks. So we wanted to see if the Python-based control software that we have developed, if it's able to control pH and conductivity after that. So we found that yes, our method is robust and it controlled pH as well as conductivity in all the cases. The last case study was to use Raman spectroscopy for online monitoring of critical quality attributes. Usually for these critical quality attribute measurement, people use HPLC. However, these HPLCs are very time consuming, and sometimes the sample preparation that is required for running these HPLC, they might also hinder with the CQAs. They might also increase your aggregates, as can be seen in some of the publications. The two critical quality attributes that we took forward for this are charge variants in case of monoclonal antibodies, as well as aggregates in case of erythropoietin and human growth hormone. So usually charge variants, they are usually separated through analytical cation exchange chromatography, and aggregates, we usually see this in size exchange chromatography. But even if you want to shorten this method, it will take you roughly 5 to 30 minutes, and then you'll have to analyze those products. So this cannot be a good tool if you want to implement this as a real-time tool for continuous chromatography control. Thus, we decided that we'll use Raman spectroscopy for controlling these critical quality attributes. So the methodology is, first you prepare some samples, you run HPLC, and of those samples you run Raman spectroscopy. Then you develop a model, and then you validate that model. Once you have a model in place, you can now use it in continuous manufacturing. The first CQA, which is aggregate determination, it was done on EPU and HGH. So we took multiple samples, we kept it at high temperatures intentionally to make some aggregates, so that we have a lot of variety of aggregate percentages that we can incorporate in our calibration model. The better the calibration library that we have, the better the model it's going to perform. Once we had all the samples, we performed HPLC, same SEC HPLC and Raman spectroscopy to correlate both of them, and we developed a PLS-based model, and we had a good model with good R-square values and low RMSE values. The next CQA was charge variant. For charge variant, so for aggregate it was easy. You just need to put your samples at high temperature for various amounts of time. But for aggregate, the sample preparation itself is very difficult, because in case of monoclonal antibodies, there are multiple charge variants that you can see, and how will you decide how to pool these? So we performed semi-preparative or semi-analytical chromatography, which has higher resolution. After performing this, we pooled all of these samples. So this is your acidic pool, this is your main pool, which is your main product, and this is your basic pool. Ideally, you would want to have 100% of this main pool, but there has to be, since this is a semi-preparative scale and you can see the resolution is not that good, so you have to have some sense that how much percentage of acidic or basic are allowed in that sample. So once we had purified these acidic main and basic species, we mixed them in different ratios and we developed some models, and we found that CNN model was best when it comes to acidic main, basic and total species determination. Then we validated this model. So we ran a different chromatography. This is a preparative chromatography where you can see the resolution is not that good as compared to your semi-preparative chromatography. We collected each and every fraction, and in that each and every fraction, we did Raman spectroscopy and correlated it with HPLC, and we found that both of them were correlating very good. So there is close to little error that you can see from this validation experiment. Thus, you have replaced a state-of-the-art HPLC, which used to be a gold standard, and you have replaced it from HPLC to Raman spectroscopy, and now you can do these analysis within a few seconds. So where you were doing it in 5 to 10 minutes, and you need to have some time to make a decision. Where this continuous chromatography, all of your runs are happening in continuous mode, you have no time to make some decision. This will give you that leverage. So to summarize, process control is very important if you want to have a continuous manufacturing system, and thus this will help you in decreasing the cost of biotherapeutics by enabling the continuous manufacturing. And the process control is actually a combination of different PAT tools, different models, or efficient digital tool platforms to ensure that there is long-term stability into your end-to-end continuous manufacturing platforms. With this, I would like to thank my research supervisor. I would like to thank the DBB department for considering me for TK Ghose Memorial Award. And I would like to thank my lab, Center of Excellence Biopharmaceutical Technology. Thank you.
Thank you, Dr. Nitika. Let me take the seat.
Thank you.
Thank you. Our second awardee is Dr. Shantanu Banerjee. He will be joining us online today. Currently, he is working as advisor at Eli Lilly and Company. He is awarded for his work on process analytical technology-enabled monitoring and control of biopharmaceutical operations. Dr. Shantanu was jointly supervised by Professor Anurag Rathod and Professor Anupam Shukla. I now request Professor Anurag Rathod to introduce his work to us.
Okay, so I'm sure everyone today has heard of AI/ML, right, and how artificial intelligence and machine learning apply to pretty much everything we do. Shantanu's thesis is more on how we can apply advanced data analytics towards essentially what Nitika presented, but from a modeling and data analytics perspective. So I hope you'll find the two sides. I mean, Nitika is more of a experimentalist and Shantanu is more of a modeler and I would say process design. So hopefully you enjoy. Thank you. And again, thanks to DBB for the award.
Thank you, Professor Rathod. I now request Dr. Shantanu to deliver a short presentation of his work done at IIT Delhi.
Good afternoon, everyone.
Yeah, you are visible.
Am I audible? Can anyone confirm?
You're audible.
Oh, thank you. I need access to share the slides. And please form with access as well as you.
Say if you can check now.
We can do that. Thank you so much. Please do the work. Yes. I hope my screen is visible. Sorry.
Yeah, it's visible. You can start the presentation.
See, thank you so much. Thanks to Delhi Department. Thanks so much, Solution Committee, specifically Bush. So, I think one of them with this award and Solution is one of the professor of this. So, I have been working on production. So, those papers implementing and this was towards upstream and harvest process, where the focus was to towards enabling the information process. And, so my main focus has been to work with controls in Yaman and also in this technology, in this step wave separation. So starting with the PAT, this is the system which can design reason to improve manufacturing. So the idea is to measure you can do is you can monitor consist parameters and then use those models in a way estimate CQV and then over a design process where we can get the optimal outputs for the process. So basically, in a way, we need to just have good measurable for the CQV and which can give us a good system for the CQV.
Dr. Shantanu, your voice is cracking. Do something to your mic. Do
Do something to your mic.
Sorry, is this clear? Is my voice now?
Is it better now? Is it better now, audience? No.
Okay. Okay, do let me know if there's silence from bottom. So, now,
Shantanu, please use your please use your system's mic.
I am open now. Can check if my voice is clear right now? If I'm audible? Okay. So I'll try.
Okay, try.
The microphone is working from my end.
Yes,
Is
it's
it clear?
better now.
Am I clear?
Yes, it's better now. Proceed.
Okay. Yeah, sorry for the glitch. So, moving to the the PAT. So we have all our processes from upstream to downstream and the formulation. And the idea here is that we need to have these real-time the tools which can capture the information, the data from the processes, be it the bioreactor or the chromatography or the filtration, using spectroscopic tools like NIR, Raman, and then we need to have a cyber framework where we have these in silico models. It can be a mechanistic model, or it can be AI-based model, or a hybrid model. And these models will give us the real-time estimations for the specific process. And then the idea is to implement real-time control as well on the process. So then the model sends the activated values of these parameters, and then the result is that there is a closed loop, and we are able to control that specific unit of operation or a deviation which is occurring. So the idea here is that we can use PAT to enable the process from batch to continuous, as well as we can also try to minimize if any deviation will propagate from one unit operation to the other. So the first case study for my work was to develop an IR-based tool or a spectroscopic tool for the CHO cells and the E. coli fermentation. Previously, we had been using HPLC to estimate all the amino acids, the sugars, and so this basically had a lot of the analysis time of more than an hour or two, and what it led to was that it was difficult to use the values of the concentration values of amino acids and estimate what is the current state of the E. coli fermentation process or the cell culture. So the idea here was to develop in silico and a model where we can have the data in real time and we can estimate using spectroscopy, and we can get the real time of these analytes within the bioreactor. So the challenge what it served us was that for NIR and Raman, we have so based on amino acids, we have multiple peaks which kind of overlap, and this poses a big challenge to the model and a particular spectra, because each spectra will contain a plethora of information from all the metabolites, sugars, as well as the inhibitors. So the idea is to have a very robust chemometric model which can find out the exact amino acid concentration which we are trying to focus on. And generally, these amino acid concentrations are always less than they're in the range of around 1 to 8 millimolar. So the main challenge is that the concentration is very low and the signal from the other molecules is pretty high. So the idea is to have a very robust model which can give us a prediction in order to estimate these low concentration molecules and get a overall good prediction. So to do this, we basically need a very good feature selection, data pre-processing, and an NVDA model which can give us a good quantitative and qualitative analysis. And this can help us to make the perfect model for a PAT framework. So this is just how a spectra looks like, where there are multiple values, multiple bonds, and the idea here is that the overlapping has to be understood for like 20. If there are 20 amino acids, so the idea is the model should be able to understand the specific regions of the spectra and basically evaluate the changes because there are the bonds are common for multiple amino acids. So the idea is to have a very key understanding of the change in the concentration and the respective bonds which are linked to these amino acids. So I will directly move to the results in the interest of time. So what I got from using the convolutional neural network models was that these models were able to give me a very good estimate with almost all the amino acids and also for the acetate, glucose and lactose in such a way that it always, the prediction was more or less above 90% for most of the batches which we use. And the impact of this is, and again, this study involved multiple pre-processing for the data set. The overall impact you see is that now, if for an E. coli process, where we know that the doubling time is very less, here within a minute, I can estimate the concentration of the amino acid, and eventually I can also implement real-time control as well. Previously, which was not possible because it used to at least take an hour or more to get the value from the HPLC. So if now in our case, we were able to estimate within a minute the real-time values of the amino acids, the sugars, and this enabled us to make a real-time control model for optimizing the amino acid concentration so that we can increase the protein productivity and the yield. The similar thing was done in CHO cell culture also, where we used these models to monitor in real time, and the predictions were pretty much accurate with more than 90%, and the overall root mean square error was less than 0.3 millimolar, which is in a way a significant achievement because for concentrations less than 1 millimolar, so having this less error basically leads us to even monitor these molecules at very late stages where the concentration dips very low, and then even then the model will give us a very good estimate. So in a way it enables the upstream that you can monitor even the very minute of the metabolites or the sugars very effectively. The overall impact, if to summarize, is that using an HPLC, we are having a time of more than 1 hour, and in case of using these chemometric tools and the spectroscopy like NIR or Raman, we are able to do it within a minute, and basically it leads us to enabling the upstream to work more efficiently towards process control as well. So next part of my study was on a novel harvest technology for CHO cell culture, which is acoustic wave separation. So basically what it does is that it uses three-dimensional acoustic waves which kind of capture these CHO cells within the nodes of these waves, and eventually what happens is that these cells tend to cluster and settle down as form of agglomerates. So it's like a filterless technology. You do not have any membrane, and you are trying to separate the cells based on these acoustic waves. And this, the best part is that this instrument can work in a very continuous fashion, and it has very less heat dissipation, or it does not impact the monoclonal antibody quality attributes. So it can be implemented in as a harvest technology for the CHO cell manufacturing, for the trastuzumab production which we use. So the first task for me was to develop a mechanistic understanding of how this agglomeration is happening. So what I did was to apply a population balance modeling, and we evaluated the overall effect of all these forces, the acoustic forces, which were leading to the cells getting agglomerated. And so now, after the study, we understood that what equations and how much, which parameter was leading to a higher agglomeration, and this basically gives us a good understanding that even if we change these parameters within the design space, we can achieve a higher cell separation as well as a higher protein recovery, because the idea here was to maximize the liquid fraction, the liquid fraction retention, because within this liquid, we have the monoclonal antibody. So the idea was to maximize these cells at the bottom, which were settling down, and recover the liquid as much as possible, the supernatant, which basically contains our product, the MAM. So then we have estimated which parameter were being critical the study, how the field flow rate basically is, leading, if a higher field flow rate is there, it will lead to a very high turbulent regime. So how this field flow rate and acoustic power and the overall trajectory of the cell were being impacted, and the net effect was that we understood that, okay, at using a optimal value of the acoustic power, a lesser feed flow rate and a higher cell density, we can achieve cell separation more than 90%. So this was the impact of doing the mechanistic study, post which we understood that, so the instrument also has multiple chambers. So we can also optimize the separation based on each of this chamber in such a way that the eventual result is more than 93 to 95% overall separation. And we can also optimize the separation within each chamber so that we can stagger them eventually at a later stage to make them into parallel modes, or even in case of like series parallel mode. Another study which I did was using the DOE approach to understand the protein recovery, which was not that, it was not very easy to do it in the mechanistic approach. So we did a data-driven model with the DOE, and we understood the protein recovery, and we got an estimator at which parameter values we can achieve higher protein recovery more than 95%. And using these two models, the mechanistic model and the protein recovery model, we made a hybrid model now, because the next instrument to the cell separation, the recovery, is the protein chromatography. So now what we want is that how can we basically enable the acoustic wave separator to integrate with the downstream in such a way that the overall flow rate of whatever is exiting, the exit flow rate, the permeate flow rate, it always matches with the downstream loading. And this was done via a framework of the hybrid model, where we used both the empirical model and model, basically get an estimate for the maximum cell separation and the maximum protein recovery. And eventually, the output of this hybrid model was sent to the distributed control system, which basically had a match of the flow rate with the chromatography and the cell clarification. So having these two models, we eventually made a digital twin where any kind of deviation which was happening within the acoustic cell separation was monitored, and each of these deviations were accounted using the different models. Even also the failures of the pump and the acoustic chambers were also encoded within this entire framework in such a way that if there is any kind of deviation in the process, the model will account it for it in real time, and it will try to apply corrective action so that the error is minimized and the deviation does not flow to the other unit operation. So again, once we have all the framework, we basically made an entire holistic framework now that we are able to control the process in real time. We are able to diagnose if there is any kind of deviation, and even some deviations which can occur in the future also. That also we did with predictive maintenance. The result being that we made an overall cyber physical system architecture for the entire unit operation, where now we are able to do the capture of data, especially the relevant, the key parameters like flow rate, the low density, acoustic power, and we are able to send it to the data historian, which is a key instrument, which is a key equipment when we have multiple data coming from different unit operation, and this basically sends the data to the distributed control system, which will basically monitor if there is any kind of deviation happening in the AWS, and it will apply respective control to basically minimize the propagation of any kind of anomaly or error which is there. So the result was that if I'm having a deviation in my feed turbidity, the cells, so if there's a dip, what the model kind of does is to monitor this in real time using turbidity sensor. Then it sends it to the DCS, which basically using these hybrid models, it will compute that what parameter can be changed to ensure that the change in the turbidity does not affect the output. So that change is done by the DCS in such a way that the overall system remains unchanged. So here in this case, you can see that if we are applying no process control, we have multiple deviations occurring in the exit of the different chambers in such a way that now the exit turbidity is more than what we expect and we don't have a clear separation. So what we do is that we apply this digital twin and cyber physical system network, and now once the system is activated, it will change the parameters. So in this case, we change the acoustic power in real time. I mean, it's not we, it's the system actually. It will change it automatically, and what it will do is that these changes in the acoustic power, they will in a way control the turbidity of all these chambers in such a way that even if there can be a major variation more than 200% of the initial feed, the output will be unchanged. So the unchanged output means that the process is operating still, the output is still within the design space, and we don't have to make any kind of deviations for the next unit operation, which can be the depth filtration. So this is the overall aspect. And the next part was also to do a multi-objective optimization, that now we have a good process control. What we did is that we tried to study the default mechanism was to do it in series, the process separation, but we also studied that if we want to increase the throughput, so what we can do, we can also have in parallel modes or in series parallel. So we did a multi-object optimization to understand that how we can maximize the cell separation out, and as well as also we can maximize the throughput, basically which leads to higher productivity and a higher separation value. So, applying the MO, algorithms, so what we got, we saw that, in using the series parallel mode, we are able to achieve a high protein recovery of around 82.61, and the cell separation also in continuous fashion, we are able to achieve around 80%. The overall time was around 10 minutes compared to a normal operation using series, which takes 23 minutes. So basically, like a trade-off we have where in a significantly lesser time, we are able to have a good separation and a good protein recovery. And we can use this to eventually, have a better productivity for the process. So to conclude, so this has been the overall, framework for my study, the methodology, where it started from model development, where I used soft sensors and in silico optimization. The target was to do the real-time process control, and the end result is that even now the models, the digital twin, it's capable of doing predictive maintenance. It's capable of, it's like a feed-forward control that before even the error enters the system, it will be able to apply the process control. And this is possible because of using the digital twins for individual unit operations. I did it for the harvest, but my other team members have been doing it for the chromatography and the filtration. And then again, everything combined together in a network using the cyber physical system enables us to use all these different digital twins to enable continuous process and real-time control of the entire biopharmaceutical operation. So that is the advantage which I realized when we worked along with TCS to automate the trastuzumab production process in our lab. And of course, AI is always there. So be it any model today, we have AI which helps us to make more better predictions over the time. So yeah, to conclude, my main objective has been firstly on the upstream where we use the NIR tool and develop this model for real-time estimation of metabolites. then I worked on the hybrid modeling, and I realized that using a hybrid model, it's more better to achieve higher, better process estimates. And then eventually, the idea is that if you have a model, the idea is to basically apply in some way that we can have a better estimate of the process controlled variables, and the result should be that the process the deviations are minimized to the maximum extent. And the last was doing the multi-objective optimization for the process to even increase the productivity or the separation further. So yeah, this is what I had done in my PhD work, and I would take this opportunity to thank my supervisor, Professor Anurag Rathore, and my co-supervisor, Professor Anupam Shukla. I also want to thank my lab members from CBT and my colleagues from TCS, so who all came together to make this project happen. And yeah, and again, thanks to the entire selection committee for bestowing this prestigious award.
Thank you, Dr. Shantanu. You may stop sharing your screen. We will now move to the award ceremony. So I now request our alumni, Professor Subhash Chand, to felicitate our awardee, Dr. Nitika. Sir, I request Professor Subhash to please share his memories of the department and IIT with us.
Thank you, Dr. Garima, to give me this opportunity to share a few words of my memories. It's a long time back. Of course, I superannuated in 2011, and but my association with Professor Ghose was also very long, probably right from 1966. 1966, I was his research student for BTech project, and then also for PhD project, for also my PhD work. And of course, I also happened to work with him as a colleague, as a younger colleague in the department. When I joined, it was Biochemical Engineering Research Center, popularly known as BERC, which gradually got shifted into a full-fledged department, Department of Biochemical Engineering and Biotechnology. Well, Professor Ghose, as I remember him, he was indeed a very sort of visionary, great visionary. I still remember the very first lecture I heard from him in 1966, when he was visiting IDPL Rishikesh as a consultant, and he was passing through HPTI Kanpur, and he delivered a lecture. And I remember in 1966, not much of biotechnology was being talked about. but he was in full steam to motivate us. The next future lies with animal cell cultures, plant tissue cultures, and microbial. They will take over the whole. Uh, Particularly, he referred to environment and biotherapeutic science. And particularly, I still remember some of his examples he chose was to produce blood proteins, blood components, blood plasma components in terms of proteins through uh, microbial cells. Although the term recombination was not, known in those days, recombinant DNA technology did not exist. But ultimately, we have seen that many of those dreams have come true. And I also remember my association in the time when he was under a great struggle for his views, because many of his views were not really accepted in the scientific community. A hardcore chemical engineer converted into a microbial engineering, and so there was a lot of difficulty, and probably I think that must have motivated him to generate a class of his own people who can understand him and who can deliver the goods. And I am sure that the two awardees this year, you can really feel that they have moved probably a few steps ahead of what the legacy he left. And of course, at the time when he was very active in research, was the use of ethanol as a automobile fuel. Even that time when he was there, when we were working in the department, in Department of Biochemical Engineering, on alcohol or ethanol fermentation based on lignocellulosics, there were a lot of question marks. But today we see it's a real reality in our own country, and the government is forcing to use 20% ethanol to be admixed. So probably that his vision has come true very fast. And as a human being, I think he was an extraordinary person. In the sense, from the outward, he was taken as a very hard person, very hard taskmaster, very tough person to deal. But at heart, I think he was very soft. And I still remember many moments when me and many of my friends who were in distress, he really gave advices which worked for us. And I just like to share, two of his students, they became so much distressed with his PhD work, then they left the program. They left the program and went to CSIR for a job. But then suddenly, I think probably within a couple of months, he called one of us, sent them the message that let them come back and complete the work. And they came, and today both of them, of course, unfortunately, one of them has recently expired, but otherwise, both of them really did wonderful work and rose to quite good heights. So with this, I think I take leave, and I thank the department to continue the program which some of my friends have started. And I think this will keep us remembering his message and his contribution to biochemical engineering, not only in India, across the world. Thank you very much.
Thank you, Professor Subhash, for sharing your memories with us. I now request our alumni, Professor A.K. Dubey and Professor Vikram Sahai, to felicitate our awardee, Dr. Shantanu. Since Dr. Shantanu is only joining us remotely. So I request Professor Anurag Thakur, his supervisor, to to please collect the memento on his behalf.
I just wanted to say so it's Anurag Rathore. Anurag Thakur is like a more famous person. No? I often get called Anurag Thakur.
Thank you. I'm so sorry for this. Sorry, I request Professor A.K. Dubey and Professor Vikram Sai to please share your memories with us. Professor, you can take this mic.
मैं 1971 में IIT में आया था to do my MTech and thereafter PhD. I completed my PhD in 1979. Throughout, I had the opportunities of interacting with Professor T.K. Ghosh, and he was really a taskmaster. He made us work like anything. He inculcated in us do hard work and be in a discipline. So he made us work in a very disciplinary manner. He would come to the lab at 8 o'clock early morning, and he would ensure, he will expect that all of us are there at our respective workplaces. 8 in the evening again he will come, and we got we got to be there in our respective places. So, as a matter of fact, he made us into someone who can contribute to the society, who can do some positive work, and eventually through his uh, through his advices, we rose to wherever we are nowadays. I really, you see, yeah, I must share one thing. Every Friday, he would call us for a discussion. Discussion means over the past week, whatever work you have done and what you are going to do. Discuss. And really, we tell you, the moment we entered his room, by the time we came out, we were shattered into pieces. And throughout the day, we will not be able to do any work. In the night, we'll see a movie, late night movie. Next morning, we would start our work. Oh, but really, I admire him. So that's what I want to say. That these are some of my memories when I was a student over here.
I first met Professor Ghose in 1983 when I was visiting the department. It was the conference, IBS conference, December, right? December 83. February 83, right, yeah. So it was during that period when I had met him, had a chat with him, and so my voice is loud enough. So I knew him before coming here while I was pursuing my master's. I knew him and I had a kind of drive, a great desire to see him. Now, heard so much him during my, and some of my senior students were here, colleagues, and they, through them and through other discussions. So after my first meeting, it was so motivating, it was kind of very inspiring. And it was then when I decided that probably I will pursue my PhD at DBEB. It was not DBEB that time, it was BERC, Biochemical Research Center. So in the same year, I got admitted to the program, and he was one of my supervisors. So I did not have that direct confrontation as you guys used to. So there were two people securing me, right? So two of my supervisors stood guard, kind of. But he was very, every time, whenever you meet him, he will catch hold of you and will ask all the details, what you are doing, how you are doing, what you want to do. And it was not only during the course of PhD, but subsequently I spent a few years in US and then came back here in 1981, joined the department as faculty. So by that time he was retired, but he was still in the department. He used to come every day. Wherever and whenever he will catch hold, I mean, he meets you, he would know everything. His passion of science, was so deep that I remember meeting him just very little while ago before his demise. It was in I think 2018, 2019, sorry. 2019 he passed away, but I think I had met in 2018, it was at Harar. We went to his home, me and Professor Ghose were there together, went to see him. He would still talk about science, and he would still talk what is going to happen, in the times to come. So he was so passionate about the science drive and what to do, where science is going and all that. He would ask us, tell me something, abhi kya chal raha hai, kya ho raha hai. So up to date. Also he was an excellent reader. He would read the literature, he will keep himself updated. So passion at that time, at the age of 93, 94, maintaining that level was remarkable. So these are kind of the memories that I have with him. Thank you very much.
Thank you, Professor Dubey and Professor Sahai, for sharing your memories with us. I also congratulate again both the awardees, Dr. Shantanu and Dr. Nitika. I now request Professor Preeti Srivastav, Head of Department Biochemical Engineering and Biotechnology, IIT Delhi, to deliver vote of thanks.
So thank you everyone. I have acute cold, so my voice is not the best. Starting with the director, sir, I would like to thank Director IIT Delhi for sparing some time out of his busy schedule. Very special thanks to Dr. Tasleem Harif Sayed, because he agreed on a very short duration, I would say, and your talk was really inspiring, very motivating for all the students. I already received messages on my mobile that the students are willing to interact, so thank you so much for a very inspiring talk. I would like to thank our senior faculty members, alumni faculty members, Professor Subhash Chand, Professor Vikram Sahai, Professor Dubey. Thank you so much, sir, for coming to this event and making it successful. I would like to thank all faculty of TBEB, all staff and all students. Without their support, I don't think this entire event would have been successful. So thank you so much. Very special thank you to Professor Hasnain, Professor Sri Krishnan and Professor Shilpi Sharma for help at multiple steps. Among staff, I really want to thank Nitin Kandpal and many other staff members who have supported. Among students, I have a list So this time we instituted some new changes in the award ceremony. We introduced a memento. So I would like to thank two of my students, Aditi Keshav and Purvika Kulshresht, for designing the memento. UG student Avinash Singh designed the standee, and a few students, I would name them, Sai Ganesh Nair, Navneet, Dev Garg. These are our MTech students and MSR students. Also Tanu Gautam, so they helped in designing the brochure. We also have Nidhi Patil, Avinash Verma, Khushi Chauhan, and Tanya Bansal, who have also helped in making this event successful. A very special thank you to some of the student representatives, Pragya from first year, Janvi Sharma for second year, Shatakshi for third year, Triveni Shelke for fourth year, and Shubham Sharma for fifth year. Thank you so much to all of you. Thank you.